Thank you very much. My name is Predrag Radivojac. I've been in this school for 14 years, and it's actually good to see some of the folks who were there when I was hired, especially Ed Robertson, who was my first faculty mentor back in the day. I'm going to tell you a little bit about research from my own group and also research from the Precision Health Initiative. And research from my own group will be limited to a particular set of activities that is related to precision health. So what you see here is on your left side is a molecule called hemoglobin. Every one of you has a lot of those. In particular, we have about at least 100 grams per liter of blood. Okay? This molecule actually takes oxygen and takes it from your lungs everywhere else and delivers oxygen, and that's how we function. So this is a protein that contains 4 different components, chains. If you pick each one of them, so they're in different colors, and you kind of pull it one end from the other, you will get a linear chain. And that chain consists of amino acids, and these amino acids, depending on their composition, this protein will form. And here we have 4 of those things that form. What happens with this protein is that if there is a variant or a mutation at position 6 from E to B, and I will not bore you with details who is who here, what happens is that this protein comes together with another protein of the same type, with another copy of that protein, and they clump together and form this sort of octamer. There's 8 chains now contained in here. And there's a structure of that solved in regular database. And then those octamers come together, and this is a simulation in this picture here, when they start clumping together somehow and form fibrils. And these fibrils, now you can see a picture of a cell, where these fibrils actually go outside of the cell membrane, and the cell itself takes a sickle shape and cause a disease that is called sickle cell disease, and one of those is called sickle cell anemia. This is not surprising that the people are anemic because of the lack of oxygen that can be delivered elsewhere. Now, of course, even though there is only one mutation here that can cause a disease in a recessive state, manifestations vary widely and basically depend on the entire genome of the person, and of course, lifestyle. Now, we have many of these projects going on. We know one of these mutations, for example, sickle cell anemia. There's about 40,000 mutations for which we know they cause disease. They were studied by geneticists. But only a handful of them, let's say 100 of them, we know actually how this happens. So one of the goals of my group is to understand how these mutations cause disease, or whether we can integrate genetic data where we can study that a particular mutation is important, and connect it with molecular data to figure out how this happens, what happens at the molecular level. Now, there's a lot of data coming together. There's Personal Genome Project, where you can find about hundreds of genomes already available at Harvard. It started in 2005, the first paper published in 2012, and now many others are popping up in UK, China, Canada, and Austria. There's Genome Aggregation Database, contains already more than 100,000 exomes, which are the only pieces of genomes that code for proteins. They also have more than 15,000 genomes. And of course, there's a lot of data from cancer sequencing projects where we have the mutated genomes that cause havoc in a person's body. There's all other types of data. How can we integrate that? How can we do something that will make us understand how diseases happen and make us also figure out how to cure them? Note that that one hemoglobin pair a lot of quaternary structures of hemoglobins when they come together. Am I there? Late. I have 10 minutes, I think. All right. I do have 10.
No, Petra has 10. Petra special. Go on, go on.
So wouldn't it be nice if we can put a little drug in between those pairs of proteins so that they don't come together, so that the person actually does not get those fibers? So our group studies proteins, and if you take a person's genome, these blue rectangles are parts that are called exons, where these mutations or variants happen. And you can see here certain proteins where particular people might have differences in their protein sequences. We are interested in those variants that have the potential to change protein structure and function, and therefore be targeted for either better understanding of disease or perhaps targeting to be cured. Now, I don't have too much time to tell you what protein function is, but just a quick story, one that calls this, you can see it here from sausages to wrinkles. You know, back in the Napoleonic times, they were noticing that when soldiers eat uncooked sausages, they get paralyzed from head down. Turns out that they had eaten some bacteria that's called Clostridium botulinum, and that bacterium secretes a protein that irreversibly binds to your neurotransmitters, and because of that, it is paralyzed. But it also turns out that, so we know what happens at the molecular level. But it also turns out that when you take a little bit of that, you can help your migraine. And if you put a little bit under your skin, well, it can take care of your wrinkles. So these proteins do many, many things. And so we want to understand what they do. For our group, we take a limited view. Our limited view is that, for example, in this particular protein, it binds an ion of zinc. This one gets covalently bound to phosphate group. Another one gets bound to DNA. And all of these proteins, all of these events can happen to the same protein. And also, many of these events can happen. How can we figure out what went wrong if there is a variant? So we have some methodology. I will go rather quickly through this. So if there is a position 35, let's say in this protein sequence where something happened, is there going to be effect to that protein if there's a mutation at position 37? The only way we can do that is to predict that. And so we develop a lot of models, and our goal is to develop a model that can say what is the probability of an event before a mutation and what is the probability of loss of that event after mutation. This is a difficult process. This has to happen in a so-called positive unlabeled framework, which is a part of semi-supervised learning. There's a lot of difficulties to do that. And so we have developed models for more than 50 of these different types of events, from metal binding to catalytic residue, loss of catalytic activity, DNA binding, protein-protein interactions, and so on, all in the positive unlabeled framework to be able to actually prioritize what goes wrong when a particular variant happens. And then once we develop all these predictors, we decided, well, let's test them. We have collaborators at UCSD who did some protein-protein interaction assays for us. And when you take a syntaxin binding protein, this is in a cohort of autistic patients where they have de novo mutations. Those are mutations that neither of their parents has. And so this is wild type protein. When you see white here, it means binds these three proteins. And when there is a mutation, you see it does not bind. And so we tested some de novo mutations, and by and large, we found out that our predictions worked. And the top 10 that we prioritized, we only had budget for 10. Actually, we got very good results. So we think that this computationally driven study of disease provides us a better understanding of these events and probably some hope how to continue in the future. Now we go beyond genetics and molecular data. Our university has embarked on an initiative called Precision Health Initiative, where we have publicly stated goals to cure couple of diseases, to prevent one chronic disease, one neurodegenerative disease, to grow our research capabilities, and also develop our educational programs. Our school is involved in only, well, across the board of these activities, but two of them are most important. One is, of course, to grow our research capabilities and also to attack the problems that computation, statistical inference, data management are challenging problems and actually fundamental for the success of precision health. This is our team. We have 6 research teams. So from variant and genome interpretation, which is one thing that I show you that my group does, to omics, data analysis and interpretation. Haishu will talk about that, I hope, rather soon. Clinical decision support, high performance computing, security and privacy. Of course, community engagement and impact analysis of our project. There's one project where so there's a lot of spawned projects from this. Some people work immediately or pairwise with our colleagues from Indianapolis. Others are working here. There's a lot of activity. But one project where we do work together is a study of gestational diabetes, where we are trying to develop a risk assessment system from different types of data in which we can actually study gestational diabetes, which is diabetes that happens to women in their 3rd trimester, but we would like to make a model that can predict it in the 1st trimester. This is important because gestational diabetes and diabetics who develop gestational diabetes fall under the highest risk group of developing type 2 diabetes 5 to 10 years after the pregnancy. So if we can develop this risk model and later target it for intervention, that would be pretty good. So here's the project. These are my last 2 slides. We have several activities. One is that we got approved by the New Moms to Be consortium to genotype 10,000 women who were studied by the NIH and collected a lot of blood specimens. And then after that, we are going to develop a cohort or boost that knowledge that we infer from New Moms to Be study and develop a prospective study where we will collect all types of other types of data, such as wearable devices. We will interview these subjects for richer information. We will study their metabolomes and microbiomes, develop a lot of omics data, and try to develop this model, and again, try to develop a model that will then result in intervention. Of course, we would like to study these subjects, both mothers and their babies, and of course, collect data that will be in the system for the future use. And just to tell you how many people are already in this project and where they come, from several folks from Indianapolis to a number of people here in Bloomington, and not everybody's picture is here, and also the project is open to others who can contribute. But this is a large study that we think will connect us and the folks from Indianapolis into a coherent group that will transfer research into medical intervention. And with this, I would like to thank many people, but on the circuit board, as you can see, are my group, who I'm really thankful for. A number of students. Vikas was the person who developed all these predictors for understanding molecular mechanisms of disease. Kim might be here. She's also now core of my group, and a number of other people who solve various types of problems. Thank you.